Hello, students. I just wanted to go over some questions from last week, which is about ecology. You can tell that we've done this unit more recently because there were fewer questions than there have been the last couple of weeks. I still want to go over just a few key ideas. So, the first question that came up was this idea of niche partitioning. So, first of all, a niche is just where an organism can live plus the job that it does in that space. And it's really controlled by two things. It's controlled by its fundamental niche, which is sort of limited by abiotic factors like temperature and water, things that this organism needs to survive. And then it also has this realized niche. That realized niche is generally a smaller possible area because there might be other organisms that are better at competing. There's this idea of the competitive exclusion principle, where if there's two organisms that have the same niche, one is going to be slightly better and it's going to survive while the other ones go extinct or in that area. Sort of an exception to that is the idea of resource partitioning or niche partitioning, which is basically when there's a group of organisms that could use the same area, they have the same fundamental niche, but they might choose to use different areas. And when I say choose, I don't mean like a logical choice. I mean something with their evolution sort of pushes them towards using different areas of that larger niche. That way they can all exist. There's this example here with these birds, where we see all of these different warblers could all live in any part of a tree, but when they're all found together, they don't. Some focus more on the top, some focus more on the bottom, and there's slightly different adaptations that they have to allow them to work a little bit better there. That way, all of these different warblers can exist in the same place, and there's no competitive exclusion. All right, the next couple of questions that people had were about population size. So, population size can be controlled by a whole bunch of things. There might be things going into an area, there might be birth, or there might be things leaving an area, or there might be death. And sort of the relationship between all of those things creates a population size. And that can be represented with a rate. It could be a rate of growth or a rate of decline. Rate is always just the amount of change over the amount of time. And what that actually looks like might be a little different depending on what type of growth we're talking about. So if we're just talking about like population growth, then if you take the number that's born minus the number that die, that might be your amount of change over amount of time. That is sort of assuming there's not organisms coming in or organisms coming out, like immigration and emigration, because those would play a role in that too. And within that, you might have exponential growth, which is when there's a whole bunch of resources in an area and growth is happening really quickly. Exponential growth, you can represent that with this letter R to represent the maximum per capita growth rate. And then you can take that and multiply it by the initial population size, and that can tell you what the rate might be. Logistic growth would be okay, we don't have a ton of resources here anymore, so the rate of increase is going to slow. And in that case, you need K, which is representing carrying capacity. And you can say, okay, how close are we to carrying capacity, and what might that do for growth rate? The particular question that this came from was about bison. Saying, all right, there's like 3,600 ish bison, and their population growth rate is 0.28. So in that case, that sort of means the change in the time is 0.28. We're assuming that would be in a year. So I can just take my initial population, multiply it by my growth rate, and then add that to my initial population to get the population the next year. All right. The next question that came up was the second FRQ on AP Classroom. It was actually also about growth rate. In this instance, we have this E. coli bacteria, and there's a mutated one, and we're trying to figure out how this influences ATP and the bacteria's ability to grow. So the first part of this question is saying, okay, how could we know growth rate of E. coli? Why would that tell us what's going on with this enzyme? And the reason this would help us is because ATP is necessary for the energy for cells. And the more ATP you have, the greater the chances you'll be able to survive and reproduce. And so we'd assume that the more ATP there is, the larger the growth rate will be. And so we can look at population size over time to see how much ATP is being produced, even though we're not directly measuring it. The second part of this question asked about like what would we need to control, and remember, whenever we're trying to control something, we want something to compare our experiment to. So in this experiment, they had these mutated E. coli, and they're seeing how fast their population grows. In order for that number to mean anything, you have to also have non-mutated E. coli and see how fast their population grows. And then you can see, okay, is the mutation doing anything different, or are they both the same? The third part of this was asking about the growth rate pattern. So this sort of connects back to what I was talking about before, where we have exponential growth or logistic growth. There's a graph here showing those two things. Exponential growth is going to be where there's a lot of resources. And in that case, a population is going to grow exponentially. Logistic growth is going to be when we near sort of the end of the resources that are available, we reach our carrying capacity. So growth rate is going to decrease. And you're going to sort of end up eventually with sort of a flat growth rate. Um, it's actually interesting. I just noticed these graphs look really similar to a lot of what's going on right now with COVID-19. We look at exponential growth initially of a lot of the cases, and then luckily in a lot of places we're sort of nearing a logistic growth where the growth rate is decreasing. And things like staying apart from each other help to reduce that carrying capacity. Anyway, connection I made. In this example, we're like, okay, we have all these E. coli. We put just a little bit of E. coli into this growth medium that has a whole bunch of sugar in it. So if there's a lot of sugar and a small number of E. coli, we're going to assume that at least initially they're going to be doing exponential growth because there's tons of resources and not very many of the E. coli. And then again, some reason for this would be, all right, there's tons of resources, not a lot of bacteria, so they'd be able to grow really quickly. The other question that people had was about the first Schoology question with the Tasmanian devils. And it turns out this is actually a really similar question as well, looking at growth rate and change in time. So this one again was about Tasmanian devils, and it's about this like pretty nasty facial tumor disease. Like there's a picture here of a poor Tasmanian devil with a little tumor, and yeah, and that population, their population is definitely being impacted by this disease. So the first part they asked us is, okay, before this disease happened, how was it that there was a stable population? Like, why wasn't it exponentially growing? Why wasn't it exponentially decreasing? Like, why was there a carrying capacity? And the reason for carrying capacity is generally density dependent factors. So there's sort of two ways we can look at things. We have density dependent factors and density independent factors. Density dependent factors are basically any sort of features where as the population goes up, then the force of that factor goes up as well. So things like food. If you have a ton of organisms, then the amount of food that you have will limit the organisms more versus if you just have a small amount. Same with shelter. If you have tons of shelter and a really small population, then it won't decrease the population as much. Things like toxic waste, like feces is one great example, or infectious diseases. Again, we see that with COVID-19. The closer we are together, like the higher our population, the faster it can spread. Same thing with that's a density dependent factor. Predation, stress, immigration, all of these density dependent factors are normally biotic things, like living things, that may influence the population. On the other hand, density independent factors, those are things that are true no matter what, like temperature. If you can't survive where it's hot, it does not matter if there's no one else there, like you're not going to be able to live there. Or pH, or any sort of those types of things where the amount that's there like doesn't really matter whether or not you can survive or not. In this particular case, again, it'd be those density dependent factors that keep the Tasmanian devils balanced. The second part of this is asking, okay, if our population goes way down, what's going to happen next? And the big thing that might happen is we might end up with a bottleneck. So if we have a whole bunch of genetic diversity and then almost everything dies, then the small number that's left is going to be the only genetic diversity that moves on to the new population, even if that new population is able to grow exponentially again or is able to increase in population. So whenever we have a large population that goes way down, then one of the big issues is that our genetic diversity goes down too. This last one is sort of saying what's going to happen next if we get rid of Tasmanian devils. And it turns out Tasmanian devils are a keystone species. And keystone species is going to be this really important species, generally sort of at the top of the food chain, where it has a larger than average effect on the population as a whole. So there's an example here with the shark. And the shark happens to eat these rays, and the rays sort of don't eat all the other things too much, and there's a lot of diversity, and it's great. If you end up with the sharks disappearing, then not only does their food increase, but everything that their food eats decreases. And so you end up with a whole lot less diversity overall. That's sort of what we would impact here. What we would expect here as well with the Tasmanian devils. It might collapse, might be destabilized. Some might increase, some might decrease. The diversity of the entire community would then decrease. And again, the reasoning why is because these organisms have a really large impact on the food chain, the food web as a whole. Otters are another great example. When otters are there, then they eat the sea urchins and the kelp can be there and lots that's the, environment for a lot of organisms, the habitat. And when the sea otter's gone, then there's way too many sea urchins, the kelp is gone, and there's no habitat for a lot of other organisms. So Tasmanian devils are another example like that. And that, students, was all the questions that came up this week. If there's any other things that you have questions on, please let me know so that we can go over them. Thanks.